Okay, welcome back. Thank you to those of you who've reached out, done some work, emailed, done discussion posts, or logged into office hours. If you are watching my videos but you're not communicating with me in any way, please do so that I know that you're out there and that you're doing okay. Um, yeah, this week we're going to continue into the realm of biotechnologies. So if we go over to the module for this week, the first task you'll have this week is to watch this video. So you're already doing that, getting started. There will be a few questions to answer about the video, very short. Task 2 will be to do a case study about a man named Earl Washington and his trials, I guess. That's what I'll say for now, so you don't know what's coming. So the way this will work, this is a PowerPoint form, but it's PowerPoint, not Google Slides, so you wouldn't be able to edit it, I don't think. So I took the questions from straight out of the case study and put them here. So if you could just answer the questions, I think Harley just stole my video pillow, but if you could just answer the questions here in this doc as you go, that will work. And then task 3 will be another case study, which goes through a case of someone potentially using stem cells, and we'll talk about those here in a second. This one is a PDF form, and again, I just put the exact same questions from this document here in a format that you can easily answer them to turn them in. So yeah, let's get started. We're going to talk a little bit about biotechnology today. So one of the easiest ways to look at the way that biotechnology has impacted our current world is by looking at foods. So I'm just going to click here on a picture of a natural banana. So this is actually what a real natural banana looks like. You go to the supermarket today, you see bananas like this over here, this picture here. However, a natural, unmodified banana looks like this. So over the years, scientists, farmers, have grown bananas, selectively mated bananas, selectively changed, genetically modified them to make them be larger, so we have more food in one banana, less seeds, which you wouldn't even, like today you eat a banana, you wouldn't even think it had seeds because you can't even feel them. But if you bite into a banana or just break it in half, you can see tiny little seeds in the middle. However, naturally, bananas have tons of seeds, but they've been modified now such that there's more food and there's less seeds. So they've been modified for our human benefit. And if you look over here, this is like it says what 9,000 years of breeding is done to different crops. These are size comparisons. So natural corn used to be very, very small, but now we've breeded it selectively such that it's much larger, more ripe. I mean, the difference is drastic. Same thing with watermelon. The size of this watermelon to the watermelons we see today, that is a crazy comparison. If you go on down, the same as with peaches. Almost anything you buy in the store. I mean, this was 4000 BC, of course, but look how much it's changed over the years through selective breeding and genetic modification. Another way that we see biotechnology today, I didn't actually pull this one up in tab because we just did it this past week, is with gel electrophoresis. So again, gel electrophoresis takes the way it actually works to actually lead up to the gel electrophoresis that we did in class is you have a small DNA sample which you then have to amplify using cloning. So you clone it and then you put it into it's in a plasmid or a certain type of vector, make copies of it through cloning, and then you use restriction enzymes to cut it up and then put it in a gel. So there's much more to gel electrophoresis, and we'll get into that a little bit in this case study. This case study will go very in depth with gel electrophoresis and how it has been used. So a little bit more about current day biotechnology. Here's one that's very realistic. This is actually very recent in the New York Times. You may have heard of it. Using blood plasma from coronavirus survivors and then injecting it into current patients that are suffering from the coronavirus. So the theory behind this is that once you have the coronavirus, once you have recovered fully from it, you will then have antibodies in your body to fight the coronavirus, making you less susceptible to getting it a second time. Current ideas based on other coronaviruses, which cause just normal colds, are thought to give you immunity for a few years, potentially. So they're looking into harvesting the plasma, which is found in the blood of people who have recovered from the coronavirus, and then injecting it into people that are having severe effects or severe breathing problems from the coronavirus currently, to give them that immunity. So that's another way that they're using it. This again, New York Times, March 26th, if you wanted to look this up. Pretty cool article about what they're looking to do. Hopefully, I think they're starting to do it in New York. New York has had approval to begin this process, to see if they can give any antibodies to people that are currently severely suffering from the coronavirus. So that would be really cool. Other ways that technology is being used, using stem cells to help repair nerve damages from any different types of nerve injury. Most nerves, or all nerves in the body, I can't think of any off the top of my head, that they can't regenerate on their own. So once you damage a nerve, that's it. Then that's why paralysis lasts forever. If you injure a nerve in your back, you're going to be paralyzed from that spot down if you injure the spinal cord. So they're looking at using stem cells to help regain function in people with nerve damage. I don't know if this has successfully been done quite yet. If it was, it would have broke headlines. But they're looking at seeing if they can help repair. I mean, they've done it on a small scale, as you can see here, but they're trying to do large scale. Can we use stem cells to help people with nerve damage? And this would be very useful for people who, say, are quadriplegic, paraplegic, or have spinal cord injuries and are paralyzed. Stem cells are unique in the fact that they can become any type of cell. So stem cells are cells that haven't been differentiated yet. They could become a heart cell or a nerve cell or a brain cell. They could become any type of cell. So they're trying to get these stem cells to become nerve cells to repair ones that have been damaged in people with nerve injuries. They're trying to use the same type of idea with stem cells to help regenerate. This is talking about lower limbs. They haven't actually successfully done it with human limbs. They're looking into regenerating limbs. But it has been done with small things. They've made bladders, so regenerated bladders in individuals who need a bladder transplant. Or they've made airways or tracheas in the lab using regenerative medicine. So I'm going to leave you guys with, this is a quick video about regenerative medicine.
You're looking at a few hundred cells here that are working together.
The heart muscle cells that Dr. Tim Nelson views highlight recent advances into regenerative medicine.
The simplest way to explain that is it's the opposite of degeneration.
Tissues in your heart, joints, and other areas can degenerate or break down with time or disease. Regeneration is the renewal of those tissues, which is something the body does naturally.
So one strategy is to try to find ways to improve the healing of your body, and another strategy is to actually supplement or augment the stem cells in your body so that we can improve the healing by transplanting stem cells into it.
Dr. Nelson and his colleague Dr. Andre Terzik use stem cells in their research because stem cells are responsible for growing new tissue.
So stem cells just means that they're seeds that can grow into many, many tissues.
Stem cells can come from a variety of places: embryos, which are not generally used anymore, umbilical cord blood, adult blood, or adult bone marrow.
So the type of stem cell will dictate how many different types of tissues can emerge out of it.
Scientists can engineer stem cells into the type of cells they want. Here's how it works. Cells called fibroblasts are removed from a patient's skin. They're reprogrammed into what are called pluripotent stem cells. Those cells can be taught to become any type of healthy cells, such as these heart muscle cells. The idea is that the newly engineered healthy cells, when introduced to, say, those of a failing heart, will help restore or regenerate the function of the unhealthy cell.
This is one cell that's contracting, is working with many cells, and that gives the whole tissue the contraction pattern like a normal heart.
It becomes much more real when you have a personal connection to a disease or an illness where we don't currently have good options. And this is where people are asking more and more of the questions, what about stem cells?
The answer is researchers, such as these at Mayo Clinic, push forward to make regenerative medicine a reality for patients searching for successful treatment and perhaps cures. For Mayo Clinic News Network, I'm Vivian Williams.
All right, so that's just a little bit about how biotechnology is used today. So again, after you watch this video, there will be a few questions posted here on Canvas. And yeah, that's all I have for you this week. Next week will be spring break, so enjoy that.